University of Health Sciences with Punjab. He did his B.Pharm from Delhi University in 1996 and from 1998 from Punjab University, Patiala. Later, he did his PhD from GBTU, Lucknow in 2012. Dr. Chawla has more than 23 years of teaching and administrative experience. He has credited with more than 65 review papers in national and international journal of high repute. He has also published one book and 21 book chapters. He has guided 39 M.Pharm students and is currently guiding 3 PhD scholars. His area of research includes colloidal W systems, SLNs, solid lipid particles, microspheres, etc. He has been associated with a number of international journals as a reviewer. He has chaired many scientific sessions and delivered invited talks in a number of national and international conferences. He has served as a subject expert at KUMAR University, Nainital and Punjab Public Service Commission, Patiala electoral selection process. He is a member of Board of Studies, Faculty of Medical Sciences and Academic Council at Polymer University. Years. Limited. His main area of research include design and synthesis of small molecules or compounds for anti-infective and derivatives. Dr. Sharma has more than 100 publications in various reputed journal in that in terms of national and international. He has successfully supervised scholars and 5 scholars are pursuing PhD under his supervision. He has guided more than 25 M.Pharm research projects also. Dr. Sharma has an outstanding achievement of being included in top 2% scientists of the world by research carried out by Stanford University in USA. Recently, he has selected for Researchers Link Scheme by British Council under Newton Howard, University of York, UK. He has awarded with Fast Track Scheme for Young Scientists from DBT SERC. So we welcome you, sir, and at the same time, we are thankful to have you in our panel as the chairperson of the technical session, day 2, Ibro Spotted Meeting and Workshop. And sir, this is the time now to introduce the speaker of the day, none other than Dr. Umesh Gupta. He is a dynamic personality in the area of technical science and technology, and he is from Central University, Rajasthan. So he's a young, dynamic, and energetic researcher, as we have seen a lot of achievements to his profiles and from various sources. So I welcome you all, Dr. Umesh Gupta, in the technical session of the Spotted Meeting and Workshop.
Thank you.
So Dr. Umesh is Assistant Professor of Pharmaceutics at the Department of Pharmacy, Central University of Rajasthan. Currently, he is working in the Department of Pharmacy in the area of technical science and technology. He did his PhD in pharmaceutical sciences from HS Ford University, India, the mentorship of Professor N.K. Jain. He has recently awarded with AAIPS Distinguished Young Educator and Research Award 2019 at AAPS 2019 Farm Site 360 at San Antonio, Texas, USA. He is also a recipient of First Chancellor's Award for Best Faculty 2018 at Central University of Rajasthan, Ajmer. He has been also awarded DAD Research Stage for Academics and Scientists 2017. This is a German program, DAD, at Leibniz Institute for Polymer Research, Dresden, Germany. He has the past experience of as a research scientist at Ranbaxy Research Laboratories and postdoctoral research associate at South Dakota State University, USA. He has more than 10 years of research and academic experience. Moreover, the area of research is mainly focused on Dendrimer mediated drug delivery systems, solubilization and targeting. He has also explored the area focusing on neurodegenerative disorders or diseases such as Alzheimer's disease. He has more than 78 publications, 3 edited international books, 18 book chapters, and 1 patent published to his credit. with total citation of more H index of 34 as per Scopus and 35 as per Google Scholar. So, sir, I welcome you in the technical session again, and I request you to please go ahead with your presentation.
Thank you so much, Dr. Charan Singh, for inviting me. I would first like to thank uh, the management and administration of ISF College of Pharmacy. I would like to thank Professor G.D. Gupta sir, who is also my mentor, and I always take advice from him in my is related to the part of discussing about dendrimers. The another part is of course related to its applications, how this carrier system, which we call palmam dendrimer, polyamide dendrimer, can be utilized in delivering drugs to the brain, and how it is helpful, how it can be in future. Now, that it is an irreversible progressive brain disease that slowly destroys the memory and thinking skill in a very local terms, and it is a neurodegenerative disorder, and it is the most common cause of dementia among older people, especially beyond the age of 60-65. The symptoms are generally observed, can be seen. And this disease, Alzheimer, was named after Dr. Alois Alzheimer in 1906. And nearly 44 million people have Alzheimer worldwide. If you see, this is a general data I have taken, and or a related dementia is there. And only 1 in 4 people have been diagnosed with exactly it is AD or not. Because as I mentioned that it is one of the form of dementia, but it can be Parkinsonism, it can be cerebral palsy, it can be Alzheimer. I mean, depends. So 1 in 4 have diagnosed mostly with AD, but it is an, we say always it is an old age type of dementia. Now, the challenge of treating this particular disorder is delivery of drugs to the brain. Being a pharmaceutics person, I will most talk about the formulation aspect, how we can actually get better delivery into the brain, and how we can utilize this carrier and its key advantages in delivering especially anti-Alzheimer drugs into the brain. Now, we all know that the brain is surrounded by meninges, right? 3 meninges are there, and which protects the brain. Of course, the skull is there, but before skull, the brain and CSF, then meninges, and this whole part of the body, we always say it is jacketed. It is protected by skull, uh, and a very restricted entry of the molecules is allowed through regular circulation, through blood circulation. And we all know, we all have an idea that there is an existence of blood brain barrier. Now, delivering directly drugs to the brain can be highly challenging. We can't inject through skull. Obviously, we need to either go through intrathecal injections, either we need to choose some carrier which can bypass or which can cross the blood brain barrier effectively, taking the drug along with it. Several has been reported in the past, and this is also one carrier which I'm going to discuss. Now, the requirement always with the blood brain barrier crossing is the size, molecular weight, and there is a restricted size, restricted type of molecules which can actually cross blood brain barrier. So why not, if we can bypass it, or if we can design certain carrier system which is small enough which can be which is able to cross the blood-brain barrier. And so a lot of nanocarriers you will see in the research, and are simply drug delivery vehicles. Like tablet is a dosage form which carries the drug, so as the nanoparticle is a dosage form which carries the drug. However, the whole uh, characterization, the whole method of preparation of nanoparticles are entirely different from tablet. Even administration sometimes needs a different skill, a different requirement is there. So yes, both are carrier system, both are drug carrier system, but they have a different requirement, they have a different implication also. But in the recent times, we come to know, and we know all, that the novel drug delivery carriers have highly impacted in the sustained and controlled drug delivery, as well as in the targeting of drugs. There are several carriers starting from liposome to micelle to dendrimer, to very new like mesoporous silica nanoparticles, gold nanoparticles, carbon nanotubes, very recent I can say. So, if you talk about the type of research in the drug delivery carrier system before 2022, if you see, it has shifted into nano. Now we know nano suspension, nano emulsion, nano particle, nanospheres, nano capsules. So everything is getting reduced as far as drug delivery carrier is concerned. Size, at the last, you will come we come to know that size plays a very key role in delivery of drugs. And if the drug carrier system is of lesser size, then it can be more advantageous in many terms. Now, so what is different in dendrimer? So nanoparticles are already there, liposomes are already there, micelles are already there. What is new in dendrimer? Some of the slides I have taken just to correlate this topic, just to convey this topic, that this is more beneficial compared to others also. Now, this is basically a monodispersed carrier. It is not an assembled carrier like a micelle. In case of micelle, the surfactants are assembled to form a micellar structure beyond critical micellar concentration. So if a drug is insoluble, suppose in water, and if it is being solubilized using micelle, then if such injection is injected into the body of a patient, it is getting exposed into 6 to 7 liter of fluid in the body or blood in the body. It may happen that the critical micellar concentration may come down and the micelle assembly may burst, and it may lead to burst release because it is an assembly of surfactant. Compared to that, dendrimer is a real molecule. It is not going to disassemble. It is not at all assembly also. It is a real polymer. It remains monodispersed, and dilution is not going to have any effect on the integrity of this system. Now, this is just one advantage I have taken. I'll discuss certain more, and then we will come to the research which we have performed. Now, what are the different components of dendrimer? It starts from a core, and then there are different layers or branches, and then the terminal functionality. If you typically see the structure of a dendrimer, you can easily see this is a very indicative structure of dendrimer, basic dendrimer structure, you can say. The center, the circle at the center, the violet one, G0, where it is written, you can consider it as a core. Now imagine in your mind a molecule like ethylene diamine. The formula of ethylene diamine is simply CH2CH2 and both side NH2 NH2. You can imagine this as a core of dendrimer. Now imagine that the NH2, both the NH2 of ethylene diamine are again replaced by acetonitrile, that is CH3CN, and then basically again they are being hydrogenated or simply reduced. that is, they are getting converted into CH3, CH2, NH2. So one NH2 is getting replaced with 2 molecules of acetonitrile, and again 2 new NH2 are being created at 1 NH2 of ethylene diamine. So earlier, when it was just 1 molecule of ethylene diamine, it was having 2 NH2 at both the terminal. And now with some chemical synthesis process, we have created a structure which is having ethylene diamine as core, and now a branch having now 4 NH2 at the terminal. And again, these 4 NH2 are being converted into 8 through synthesis, then into 16, then into 32. And that's how the branches of dendrimer increases. G0 is the core, and then branches starting from G1, G2, G3, and G4, or beyond G5, G6. And these we call generations of dendrimer. One layer of the interior, or one layer of the branches, when it is attached to the core, we call it as one generation, and then second, then third, then fourth. Now, why I am telling all this? Because we have a real structure. I'll show you in the coming slide. This is the exact structure of palm dendrimer, the realistic structure, or you can say the real structure of polyamidoamine dendrimer. Whatever I have just explained, you can exactly see in this structure. If you focus on the center, there are 2 N molecules, CH2, CH2, NN. And this NH2 is again converted into 2 more NH2, and the another one again into 2 more NH2, that is 4 NH2. And then again, the 4 NH2, 1 H is replaced with another, amine group, and it grows so on. It's growing like a tree. In fact, the dendron in Latin meaning is having a tree-like structure, right? So we have learned different polymers like linear polymer, branched polymer, grafted polymer, natural polymer, we say biodegradable polymer. If you remember our bachelor's day, or when we were taking the basic degree education, we come to know that there was a type of polymer which was called as a star polymer. Now, star polymer and dendrimer have a difference, that the monodispersity, or the branches, grow in a very regular fashion in dendrimer, which is not possible in the star polymers. So this is a different kind of polymer. But what is it? It is a polymer. Basically, dendrimer is a polymer. What kind of polymer? It is a tree-like polymer. What do you mean by tree-like polymer? It is having a core, having different branches and different terminal functionality. This was just to explain like what is exactly dendrimer and what kind of terminology. When we purchase dendrimer in the market, we say that I want a G3, generation 3 type of palm dendrimer. That means we need a dendrimer which is palm, of course, but having 32 NH2 group at the terminal, at the surface. So why I'm telling all this in this such explanatory way? Because we need to understand that there are different terminal groups present on the surface of dendrimer, and these surface amine groups can be actually utilized for the conjugation of the drug, for the conjugation of the targeting ligand, antibody, diagnosing agent, right? Even sometimes even drugs, right? And hence this molecule becomes different in the way that if you talk about liposome, if you talk about nanoparticle, in most of the instances, we used to physically encapsulate the drug, right? And this is a very important thing, that we generally encapsulate the drug. We fabricate the drug during the preparation of liposome, either while we are adding cholesterol or phosphatidylcholine. We used to add the drug in one step. We add the drug. We never used to conjugate the drug or simply covalently conjugate the drug, which is possible with the dendrimer. We can chemically conjugate the drug like a prodrug assembly, and that is what we have performed in our studies. Now, as I already mentioned, what do you mean by dendrimer generations? I'm not going to explain now, because we have already discussed that G0 means core, G1 is a one branch, then G2, G3, and this is the terminology of the Dendrimer. If you go to purchase the Dendrimer, you need to tell them which generation you want to purchase. Now, what is the ground reality? As I am explaining, is there anything which is there in the market, or simply we are just boasting about the key advantage of the Dendrimer? I can say there are more than 10 to 12 products which are there in the world market. Some of them, like I have taken Viva Gel in this slide. There are Flustat, which is also a gel based on Palm Dendrimer, and there are many more products which are in the pipeline, which we will see in the coming 10 years. This is just to correlate that it is not just a molecule of research or research lab, but it has now turned into the market also. Now, I already told that the excellent advantage of the Dendrimer, that is strictly below 20 nanometer. If you order in in Sigma Eldritch, the commercial Palm Dendrimer of 4th or 5th generation, and if you see the material safety data sheet, or the characterization sheet, or certificate of analysis provided by them, they claim that the size of the Dendrimer is even below 15 nanometer, and exactly it is true also. Now, I told you that these have number of different amine groups, and I will tell you in the coming slides, not only amine groups are there. Now we have COH terminated dendrimers, now we have OH terminated dendrimer, now we have NH2 terminated dendrimer, and in fact you can tailor made this molecule. Now, what do you mean by tailor made? You can select any core and you can select any branch which is suitably fitting in the definition of dendrimer, and actually you can design your dendrimer. For example, I can give you one example to you, that imagine that polyethylene glycol is being taken as a core. Now both side it will have OH in the structure. You can replace the OH with citric acid. And mind well, one group, Dr. Namazi et al. From Iran reported in 2005 this dendrimer which was made by PEG as core and citric acid as branches. They synthesized the dendrimer up to 4th generation, and that paper was published in Biomaterials. So actually you can design for polylysine dendrimer. You can use poly-L-lysine as core, and you can use its branches again to design a new type of dendrimer. You can use triazine as core, and you can design a new dendrimer. So what I want to say is that if you have a good or basic knowledge of chemistry molecules, actually you can explore ammonia also as one core, replacing all the hydrogen into 3 different branches, and then emanating those branches into the new, and then new, and then new, and that's how you develop a new dendrimer having ammonia as core. You can develop a dendrimer having diaminobutane as core. So lot of dendrimer, that is why I use the term tailor made. You decide, you choose your core, you choose your branch, and actually you can synthesize a new type of dendrimer. We also did with one of my PhD student, we took the patent also, which got published recently, and now it is under examination to get granted. So it's not a molecule which is restricted only to certain core or certain branches. If you can design new, yes, there is possibility that you can design a new dendrimer. Now, what are the other advantages? I'll tell you how it is able to load the drug, encapsulate the drug, and conjugate the drug. It has excellent solubility properties. I want to stop on this for 2 minutes and will tell you that you see in the micelle, the classical micelle, if you remember, the exterior was completely hydrophilic, the interior was completely hydrophobic, and that is the reason where insoluble drug was getting encapsulated, surrounded by hydrophilic heads of surfactant assembled as micelle. Exactly same structure actually you are seeing, but it is a real molecule, it is not an assembly, and the drug is there inside the dendrimer, which are completely hydrophobic in nature, and exterior is completely hydrophilic due to presence of multiple primary amine groups which are excellently soluble in water. So hence these are highly solubilizing, very good solubilizer, I can say. Lot of drugs, more than 100 drugs are there in the market, which are excellently soluble in water. But still the attaching branch and then new branch, and that is what we call divergent approach. We can also start from the topmost surface and going down or going towards center, and from surface to core, and that is how we call it as a convergent approach. So basically, dendrimer can be synthesized using divergent or convergent approach, which you can see in the literature. Now, as I told you, versatility in this part of discussion, that there are a lot of possibilities with this molecule, that it has hydrophilic exterior, it it has multiple functionalities. So the exterior can be conjugated with number of drugs, ligands. It has interior space where drug can be encapsulated. Even you can conjugate drug also. And there are numerous possibilities of attaching different kind of molecule on the surface of dendrimer. And I am telling you, we have number of NH2. It's not only one NH2 molecule. You have 64, you have 32 in just one molecule. So you can utilize 32 molecule for conjugating a ligand. For 32 molecule, you can conjugate drug also. So a lot of possibilities, and that is why this is a versatile carrier. I already mentioned you that how this area, I told you that the wide space which you can see by my cursor movement, that this wide space, which is, you can see the blank space inside, is actually utilized for the encapsulation of drug. One such example you can see, that how this acid molecule is getting encapsulated inside dendrimer by hydrogen bonding. It is there, it can be encapsulated by ionic interaction also. It can remain encapsulated inside the dendrimer. Now, this is what I wanted to tell you about encapsulation mechanism. Now, which is the most popular class? The answer is polyamidoamine dendrimer, which has been mostly explored for drug delivery because of its key advantages, which I have already explained, which were the majority advantages of palm dendrimer. Now, other classes are also there, like polypropylene imine dendrimer is there, polylysine is there, right? Melamine dendrimer is there, and many more dendrimer you will find in the literature. Palmamose dendrimer is there. So a lot of classes and lot of newly designed dendrimers are also there, which different research groups are working upon. Now, I started with Alzheimer's, and I have not discussed much about that. And this protocol, which I am going to discuss now, because I had to tell you that what kind of carrier I am using. And now I'm shifting, towards the first protocol which we did. We all know that donepezil, rivastigmine, and galantamine are the major drugs which are used in the symptomatic treatment of Alzheimer's. Now, the cholinergic hypothesis based drug which is mostly used is rivastigmine, and it is having a mode of action that it inhibits the acetylcholine esterase activity. By inhibiting so, the acetylcholine is secreted more in our brain, especially in the patient of the brain which get deprived of, or its quantity, its secretion goes down because of elevated ACHE activity, and rivastigmine can help in inhibition of acetylcholine esterase. Now, this drug can improve the cognitive symptoms, and that's why this is one of the first line drug referred in the treatment of Alzheimer's. However, the drug is again having the challenge which I already told you, which we collectively call blood-brain barrier, and PGP flux is there, diffusion, paracellular mechanism, carrier-mediated delivery. So a lot of mechanism through which this drug enters. But if we can actually increase the overall bioavailability of rivastigmine into the brain, we thought that it may be more advantageous in the treatment of AD, and especially if we can start with using a carrier like dendrimer, it can be helpful. Now, what we thought about when we started, we thought that we will use a dendrimer as carrier. We will use lactoferrin, or because there are a lot of transferrin receptors in the brain, and we thought to use lactoferrin as ligand. We conjugated it into the third generation dendrimer. We characterized it exhaustively. And at the last, what we did is we simply, after characterization through proton NMR, through COSY, and through C13 NMR as well, we confirmed that the PAMAM was conjugated to lactoferrin. And when we conjugated lactoferrin, after this carrier was utilized for both in both ways. At one point, we conjugated rivastigmine at at the surface of this PAMAM LF, and in one formulation, we physically encapsulated this drug inside the dendrimer, inside the pure dendrimer. So at one point, we conjugated it. In one formulation, we conjugated it to the surface of PAMAM, and in another formulation, we encapsulated it physically inside the dendrimer. We characterize it for size, as you can see that the size was less in case of encapsulated one, but we realized that the size was more because of lactoferrin and rivastigmine conjugation. That too, the size was not too high, but it was 216, around 216 nanometer with a PDI of 0.2, that is polydispersity index. But the entrapment efficiency was found to be more as we conjugated the drug, we did not encapsulated it in this formulation. Now, when we perform certain basic studies like hemolysis, release, we found that the encapsulated drug, of course, was released faster than the conjugated one. But in case of the conjugated formulation, we found that the hemolytic toxicity, which was more than 30% in case of pure PAMAM dendrimer, was reduced very high, very significantly, and which was a good sign that we can go ahead with the in vivo studies. However, we also found a kind of a toxicity with PAMAM encapsulated rivastigmine, but that percentage was comparatively lesser than the pure dendrimer. So in general, we can say that this formulation in which we conjugated rivastigmine along with the lactoferrin was more advantageous in terms of toxicity, in terms of slow release also. Now, we did pharmacokinetic studies also in healthy rats, and we found that the half-life T half was around 15 hours in case of conjugated rivastigmine formulation, which was almost like 8 times higher than the pure rivastigmine. And when we did the biodistribution, which was our ultimate goal, and this is just I'm talking about in the healthy rats, then we got some exciting result that the conjugated rivastigmine formulation was having a very high concentration as far as brain is concerned, which was our main focus of study. Now, what we did after this actually, we took a mice model. This study we performed in collaboration in Assam University, where they induced Alzheimer's. Aluminum chloride was used to induce Alzheimer's, and we did different behavioral studies, water maze test. We also used scopolamine based models to reduce the memory, to disrupt the memory. And in one protocol, you will see we have used memantine also to improve the memory of mice. But we used ALCL3 to induce Alzheimer, and then we performed different behavioral studies like exploration, discrimination index, and these ORM tests to test the memory was performed. The overall conclusion, which I can explain in just one line, that the conjugated formulation was resulting into better output as far as exploration and discrimination index studies are concerned. We separated the stratum portion of the brain, and to assess the inhibition of ACHE activity, the color intensity was also measured, which represented the inhibition potential of encapsulated formulation was more compared to the encapsulated as well as the pure drug. So the stratum portion of the Alzheimer induced mice was separated out, and we took the photograph at different time point, and then the mean optical density was calculated, and which was correlated to the ACHE activity of the different formulation. So ultimately we came to one conclusion, which I'll summarize at the last, that the conjugated formulation was better, and that actually encouraged us to perform such conjugation in another drugs also. In the second protocol, we took donepezil, which is also one of the very well-known anti-Alzheimer drug. And in fact, when we synthesized the conjugate, and when we published this in ACS Omega, we got the CAS registry number from American Chemical Society for this conjugate, because this was the first time somebody have conjugated donepezil with Dendrimer, and we got a new CAS registry number from ACS. So donepezil was used in the study. We all know that it is a well-known anti-Alzheimer drug. The hypothesis was very simple, that we conjugated it with Dendrimer, and then such Dendrimer was expected to be delivered through intranasal delivery. However, we were not able to perform it, and then we injected it. At least in mice model, we were not able to perform effectively. So we tried it through in vivo, through intravenous route, and we got some exciting result, which I'll show and discuss in this. This is a synthetic methodology. So donepezil was first converted into donepezil aldehyde and then into ester, and this donepezil ester was conjugated with G4. Now we use the fourth generation. We thought that we will try to conjugate more donepezil in this study, and donepezil PAMAM conjugate was prepared. We characterize it at every step, as it is a real molecule. It is not an assembly or encapsulated one. It was a conjugate. So we did NMR, we performed C13 NMR. We also did the FTIR spectroscopy, and again the release as it was performed in the first protocol, again the hemolytic toxicity, and we came to know that this molecule is also less toxic, and in fact it is releasing the donepezil very slowly compared to the encapsulated one. So with this I can only say that this molecule again as a conjugate is more effective, I can say, though we did not perform the studies with the encapsulated, but the initial studies which we did not reported in the paper was indicating the same aspect, that the conjugation is more advantageous with Dendrimer. Again, we also performed biodistribution, in which we got the good result with PTZ and higher brain availability, higher half-life, and that's how we ended this study. Now in the third protocol, interestingly, because we had to use different drug and with a different approach, now in the third we utilized memantine hydrochloride. That too is a well-known memory enhancer, but this time the approach which we utilized in rivastigmine, because delivering memantine is a very challenging task. In fact, scopolamine induced memory model was used to study the effect of such formulation, which was based on lactoferrin conjugation and then encapsulation of memantine. And we were very much surprised to see that compared to pure drug administration, if we use Dendrimer, lactoferrin and memantine, the memory repair was significantly higher, and it was more improved than it was actually expected by us also when we were doing this experiment. So we conjugated lactoferrin again And I have not taken again the characterization part, but this we published in ACS Chemical Neuroscience. And this part of the study again explains how, because this was more toward the memory part, not the ACHE inhibition or other aspect, but we thought that can be focused on the cognitive repair of the mice, Alzheimer induced mice, and that was our focus, utilizing this as a symptomatic relief. And in this study also, we came to the conclusion that yes, the memantine release was sustained, yes, it was less hemolytic, yes, in the studies of discrimination index or exploration time, which was significantly better we got in case of this formulation. Even the AL induced increase for ACH activity, we did not see much difference, because of course memantine is not directly related to the ACH inhibition activity, and it is more related to the memory repairment, and that's how we got better results in case of behavioral study, in case of discrimination index determination or exploration time determination. So this was a different output which we got in case of memantine if we compare with the rivastigmine encapsulated formulation. But all in all, these three protocol, and with the protocol which I have not included in this presentation was related to glioblastoma. We also published one protocol utilizing temozolomide and chitosan at the surface of dendrimer, and we also got a very good result. We published that in Pharmaceutical Research, and that, because it was not in the scope of this discussion, I have not included that protocol. But in that protocol also we have got an excellent result in the brain delivery of temozolomide. All in all, what I can say is that this carrier system has immense potential. We can use third generation, we can use fourth generation, we can use NH2 terminated dendrimer, we can use OH terminated dendrimer, we can use COH terminated dendrimer, depending on our requirement. And that's how this can be explored even further with different avenues and different possibilities for delivering drugs to the brain. The another set of studies which we are now performing is the mechanistic based studies, that through what mechanism it is actually entering into the brain, either it is bypassing blood-brain barrier or only size is playing the role. However, those are challenging to us because we are not pharmacologists, or I am not a pharmacologist, so definitely I am exploring it in collaboration. But yes, it is a challenging to me to explore this aspect, that how this is going into the brain and what is the mechanism behind improving the bioavailability of drugs like rivastigmine and memantine into the brain. So with this, I would like to conclude my presentation, that these are the carriers, dendrimers, especially the PAMAM, which I have mentioned in this discussion through three different protocols, that these are able to deliver drugs like donepezil, memantine, and even drugs like rivastigmine. The one way is by conjugation, the another is by encapsulation. And I found that through different studies, in fact, we also published in the previous days the conjugated berberine and encapsulated berberine in one protocol. So we came to the same conclusion. But the challenge with the conclusion is that you need to purify the conjugate, you need to separate out the conjugated drug with the dendrimer, and definitely you need a good chemistry skill, a good team having chemistry skill. Then only such polymer conjugation can be performed. But the other good part, or you can say the advantageous or meritorious part of this, is that the drug the drug encapsulation, or the encapsulation efficiency, or the entrapment, whatever you say, is much higher than the encapsulation. So, and this is an excellent carrier for conjugation of drugs. So we thought that most of the carrier mediated drug delivery in targeting is focused on conjugating ligand and then encapsulating drug. We also performed this with mementini, yes, but we did also certain protocols with conjugation of drug itself, and it acted like a prodrug, and we are actually repeating it with different polymer conjugates also, like hydroxypropyl methyl acrylate, which is the new polymer which we are exploring for delivering into the brain. We already performed some studies, published some studies in delivering anti-cancer drug through conjugated polymeric micelle, but we are hopeful that if we conjugate the drug in a nano carrier, and if we inject it, and if it is going into the brain, the higher quantity of the drug can be delivered into brain. And that's how the one challenging task of delivering drugs directly to brain uh, can be solved up to certain percentage if this area is explored further. There is less chance of leaching out drug. There is less chance of leakage of drug because your drug is conjugated. It will take time to get breakdown by the enzymatic activity. Lot of amylases, lot of esterases, which are responsible for breaking the drug from dendrimer to release it. Yes, it will release, but it will take time, and by that time we can achieve a higher availability, higher bioavailability of that drug into the brain. So dendrimer conjugated drugs are better in brain delivery compared to encapsulation. The surface engineering of dendrimers can mask the cationic toxicity of amine terminated dendrimers, and that's how we can conclude that these conjugates are more beneficial, and we are trying to report this in the form of a of different conjugate, because each conjugation is not simply just changing the drug, and because each conjugation needs a different chemistry to conjugate. It's not just a physical mixing to encapsulate one drug with a different dendrimer, uh, because encapsulation methodology will be the same. You will mix it, you will stir it overnight, will filter it, and will lyophilize or whatever you can do. But for the conjugation, you need to explore that whether it is really possible to conjugate with the amine termination or OH, or what kind of strategy, DCC, NHS, or what kind of chemistry you can. So with this, we have published some work related to dendrimer based drug delivery to brain in the recent times, and this was all I wanted to share. This is my small team. These are my 4 research scholars who are working not all on the brain delivery, but mostly on dendrimer: Sarita, then Rakesh, then Vinay, and Sanya. And the work which I have shared today was performed by one of my pass out PhD student, Avinash Gotwal, as well as one of my MPharm student, Mr. Anurag. And this was all from my side. I would like to thank one and all who is patiently listening this session and giving me opportunity to share my work. Thank you so much, and that was all from my side. Thank you so much.
Thank you so much, Dr. Umesh, for a fantastic talk.
Thank you.
I'd like to invite the chairperson of the technical session,
Thank you.
Dr. Prabodh Sharma, for having some comments.
Yeah, yes, thank you, Dr. Charan Singh. And yeah, I will also definitely like to congratulate Dr. Umesh
Thank you,
for
sir.
such a wonderful talk and really an outstanding work he has carried out.
Thank you, sir.
Sir, your talk was really very impressive and eye-opening, and it's really a need of hour, because around 98% of micromolecules and almost 100% of macromolecules cannot cross the blood-brain barrier. So that is one of the biggest challenge. And
Yes.
Even if, despite of the potency of the drug, non-availability in the brain compartment, it frustrates the therapeutic interventions. So yes,
Sure.
you are working.
I'm sorry, my connection was
No, it's okay, sir.
lost
It's okay.
probably.
Yeah.
Am I audible?
Yes, yes.
Yeah. So,
Yeah,
Dr. Office, so my best wishes, and I am very much hopeful that you will be coming up with your own formulation in the marketed formulation very soon. So, with that, I am very much thankful to the organizers of this program and Professor G.D. Gupta sir for giving me this opportunity to attend and chair this wonderful session. And my congratulations to Team ISF also for inviting such a wonderful speaker, such an outstanding researcher. Thank you so much.
Thank you, sir.
Thank you, Dr. Prabodh sir, with the comments of the talk. Now, I'd like to have some of the points from our head of the institute, Professor G.D. Gupta. Sir, I think mic is not.
So thank you very much, Dr. Umesh ji, and I know Dr. Umesh from I think last 18 years near about.
Yes,
So and your dedication and toward the profession and the working. So today I am very happy to see you over the Ibro platform, and you are doing the lot of good work in the Central University. And here is our team of the pharmaceutics, pharmacology, chemistry. All team members, they are also the very young, and they are doing the good job in the various fields. So I hope that one of the in a form of the cluster, if we can work together, then it will be the very helpful to each other, because at a initial level, some of the one of the approach given by the some of the country, you have developed some of the product and they have provided the opportunity as you can give the molecules or give the product, they will give the proper testing, because now today is the one of the big challenge over here. At the morning our session, Professor YK Gupta ji has the rightly mentioned that nowadays we all are in the race of the factor. At the I have one of the point over here in my mind when I was just approaching some of the university at that time, when the figure is the API. So this is the one of the big figure of the UGC recently launched. This term is the API, and for the associate professor 400, for professor 500, for lecturer near about the 75. Every year required is the 50. So everybody moving toward this one. How we can achieve this of the target? But after that, I feel that nowadays this is the completely dissolved and nothing in the picture in that time. So my expectation and hope over here, what types of the research we are the performing in our laboratory, it will give the next step in the, you can add the next step in the research and give some of the future planning, so that it is the beneficial to the society. So I hope that interaction and today this types of the activity very helpful. And number of time I have include one word, mind and body and mind. So most of time I have achieved the answer mind and body. So I confuse that. Even a gentleman, how the body is, can we control the mind? So this is the essential, that if we is the sound mind, and if we have, because now we know that in those days lot of stress, lot of anxiety, so many things. That is why only you can study of the 4 5 year. One word is the yoga and meditation. It is included in the also medication system. Okay, you can take the treatment by the yoga and meditation. It means nowadays number of the person is the under the category of the frustration. So this is the big opportunity of the pharmaceutics person. You can develop some of the cool, effective, useful dosage form, so that it can be very you can say effective and useful to the person. So thank you very much, Dr. Umesh for accepting our invitation. And I have also go through your slide to slide, and I feel that this is the very smooth, and you can say जिसको कहते हैं मक्खन जैसा flow of the lecture. Nothing to be the hurry, nothing to be the go to the at the behind the level. Under the certain of the system, you have delivered the lecture. So it will be the useful to the audience, it will be the useful to the students, because number of time I feel that let us consider I am person of the pharmaceutics, if we can appear in the lecture of the pharmacology. So yesterday one lecture was the Professor S. Sharma. Know that near about the 50% of the things I am understanding and 50% of the things I am unable to the understanding. What this is the very, very important, what types of the presentation, how he did the work on the each and every slide. So personality, attitude, way of presentation. So everywhere is the learning, and the things is the available. Either you are the supporting the subject or not supporting the subject. So I hope that during this, today is the second day, and I think Charanji, this is the last lecture of today. So today, yes. So today our scientific team and Dr. Charanji organized the 5 lecture, and all lectures are very, very good. And tomorrow we have the one of the lecture, Dr. Aarti, and then we have the workshop on the zebrafish. So obviously, Dr. Umeshji, we are the working lot of work on the zebrafish,
Yes.
and we have the collaboration with some of the good institution, including the Central University of the Bilaspur, Chhattisgarh, and NIA. This is the National Institute of the Ayurveda, Jaipur. So if you have this types of the option in your university, because Jaipur is nearby to me, so we can also connect to each other, and it will be helpful to the all. And thank you very much, the resource person, Dr. Prabodh Sharma ji. I know that from the last, when I have joined the Vela College of Pharmacy, after that we are the connect with the Kurukshetra. Kurukshetra is the very popular for the 2 purpose. One is there is the one of the good and small, very, very historical place. Another thing is the university. So obviously, being the academician, first of all, we have connect with the person of the you can say academician person. At that time, I meet Dr. Prabodh Sharma. He is the very thorough gentleman and dedicated. So I welcome and thankful to you also. And another person is the Dr. Binoy Chawla ji. He is the near to the Baba Farid. And before that, I have only know the name. But when we have connect with the nearby place, then I feel that everybody is doing the good job. So I hope that this types of the, you can say, interaction, this types of the involve, this types of the presentation always beneficial to the student. Nowadays I feel that listener is very less. और मैंने कभी सुना है कि अच्छा व्यक्ति तो वही होता है जिसमें सुनने की क्षमता हो. वक्ता तो बहुत हैं आजकल, बट सुनने की क्षमता क्या है? बिकॉज एक और बड़ी अच्छी चीज है, हम वो चीज सुनते हैं जो हमें नहीं आती है, और हम वो चीज बोलते हैं जो हमें आती है. तो यू कैन अंडरस्टैंड कि जो हमें नहीं आती और हम उसको सुनना भी नहीं चाहते हैं, देन व्हाट टाइप्स ऑफ द लर्निंग. क्योंकि जो मैं बोल रहा हूँ, जो मुझे आ रहा वो बोल रहा हूँ, लेकिन जो मैं नहीं जानता था वो सुन के मुझको आ जाया है और मैं सुनूं ही नहीं, देन इट इज वेरी डिफिकल्ट. इट मीन्स गुड लर्नर इज आल्सो रिक्वायर्ड to be a excellent person. So thank you very much to each and everybody, our IT team, Dr. Charan, from morning 9 to you can say 4 p.m. over here. We are 1 to 1 hours we have the taken all lecture. So thanks to the team of the organizing committee. And thank you very much, Prabodh ji, Umesh ji, when you are in Punjab, खुले दिल से आपका ISFCP कॉलेज में स्वागत है. छोटा यूनिट है, बट बस काम करने की इच्छा है.
नहीं सर, बहुत अच्छा है सर.
Definitely it would be a privilege to visit your institution.
Yes sir,
and
yes,
we will definitely learn. और एक चीज सर आपने जो पॉइंट आउट की अभी योगा एंड मेडिटेशन की, that is really very very relevant. और इसी कंटेक्स्ट में I would like to add here that हमारे यूनिवर्सिटी में DPSRU में a center of excellence for yoga and meditation has been established, and first report has been submitted to ICMR, जिसमें कोविड के जो केसेस थे उनको योगा ऑनलाइन करवाया गया और रिकवरी रेट काफी, जो रिकवरी परसेंटेज और जो टाइम ऑफ रिकवरी है वो काफी इम्प्रूव्ड है. तो दैट फर्स्ट साइंटिफिक रिपोर्ट हैज ऑलरेडी बीन सबमिटेड एंड दैट सेंटर इज रियली डूइंग वेरी वेल बोथ इन योगा एज वेल एज मेडिटेशन. So I think that would be probably first center to have scientific relation of these our spiritual as well as physical techniques as well as that can be connected to medical aspects. तो सर ये आपने बहुत रिलेवेंट पॉइंट आउट किया है और आई थिंक इस पे और ज्यादा एक्सप्लोर करने की भी जरूरत है हम सबको.
सो प्रबोध जी, मैं यही कहना चाहता हूँ, आग से ज्यादा धुएं से नुकसान होता है. जो कहा जाता है कि जितनी डेथ रेट होती है, लेट अस कंसीडर आग लगी और आग में हाथ चला गया तो हाथ जल जाएगा, दैट्स ओवर. बट अगर आप धुएं में फंस गए तो फिर नहीं निकल सकते हो, वो सीधा दिमाग तक असर करता है. दैट इज वाई stress management is one of the most important things, and only the cooling system of the body, pure nervous system, this is the yoga and meditation. देखिए, जब आप पेशेंट हैं तो दवाई जरूर लें, लेकिन पेशेंट बनने जा रहे हैं तो इस रेमेडी को जरूर लें, तो आप कभी पेशेंट नहीं बनेंगे, ऐसा मेरा मानना है. सो जो अभी जो आज पूरी बातचीत हुई है या जो लेक्चर हुए हैं, इनके दोनों ही मोटो, तीनों मोटो हैं कि जो यंगस्टर्स हैं, दे कैन कम टू नो द रिसर्च, देयर डेडिकेशन, डिवोशन एंड हाउ यू कैन अटेंड द लेक्चर. आज मुझे बड़ा दुख होता है कि आफ्टर द प्रेजेंटेशन बाय द स्पीकर एक्ट करता है कि somebody ask the question, लेकिन ऐसा सन्नाटा छा जाता है उस समय कि कोई भी व्यक्ति बैठा नहीं है यहाँ पर. इट मीन्स इसके दो मायने अलग अलग होते हैं कि हम या तो अंडरस्टैंड नहीं कर रहे हैं या हम सुन ही नहीं रहे. तो कहीं ना कहीं जब तक एक डिस्कशन और एक भाव जो आज के यंगस्टर में हो रहा है कि वो रिड्यूस होता चला जा रहा है. सो आई होप टू ईच एंड एवरीबडी कि क्वेश्चन इज नॉट फॉर यू कैन व्हाट टाइप्स ऑफ द नॉलेज यू हैव टू द प्रेजेंट. आप नॉलेज लेने और कहीं भी कोई खामी में, एक सिंपल सा वर्ड कहूं, देश और विदेश में एक है कि जो फॉरेन कंट्री पीपल हैव द वन कंसेप्ट. आपके पोस्टर में 50 खामी है, उन्हें कोई लेना देना नहीं है, लेकिन एक अच्छी चीज है उसको वो लेके जाना चाहते. हमारे यहाँ 100 अच्छी चीजें उसको लेना ही नहीं है, जो एक खामी है उसको ढूंढ के ले जाना है. तो ये टेम्परामेंट नीड टू द चेंज, एंड इट विल बी द चेंज व्हेन वी कैन कम टुगेदर, व्हेन वी कैन डिस्कस. और जो आपने योगा मेडिटेशन की कही, आई हैव पर्सनली विजिटेड द नेशनल इंस्टीट्यूट ऑफ द आयुर्वेदा. देयर इज द 50 पेशेंट दे हैव द एडमिटेड इन दिस. तो हमने क्या हो रहा, बोले इनको योगा और मेडिटेशन से हम ट्रीटमेंट कर रहे हैं आफ्टर कोविड, क्योंकि वो डिप्रेस हो चुके थे और फैमिली में काफी लोग परेशान थे. सो दे आर एडमिटेड एंड दे आर नाउ इज द कूल डाउन पोजीशन. तो ऑब्वियसली दिस इज इफेक्टिव एंड यूजफुल. सो चलिए हम योगा की तरफ चले जाएंगे. तो डॉक्टर उमेश जी ने जो टेक्निकल सेशन लिया है वो कमजोर हो जाएगा. सो नो डाउट, पहले ही कहा है कि अगर पेशेंट है देन मेडिसिन इज कंपलसरी, और पेशेंट बनने की तरफ जा रहे हो तो इन चीजों को जरूर धारण करें, सो दैट you can avoid the so many things. So after all, today's all 5 lecture one by one, मैं शायद 4th lecture में connect नहीं हो पाया, but before 2-3 lecture and this one lecture I was the listen very carefully, और कुछ slide के मैंने screenshot भी लेता रहा हूँ, so that कि जब हमें और time लगेगा then we also do the more and more homework. So over to the Dr. Charan, please.
Thank you so much, sir, for your insightful comments or the suggestions. So far the lecture as well, the yoga and meditation is concerned. So far the lecture is concerned, sir, of Dr. Umesh Gupta. It reminded me of 2017 when I was an international participant in Ibro school at IGNTU. So I was searching the person you acknowledged in your presentation, Avinash. So, sir, I came to know about you in 2017 when I was Ibro.
Oh, Avinash, yeah, Avinash attended that Ibro school in IGNTU, Amarkantak, and he was very good in doing this study. In fact, the 2 protocol which I presented, he only performs, and nowadays he joined North Dakota State University as postdoc in US. And what Gupta sir said very rightly, that yes, it is a time that we should focus on stress management, and that is very, very important aspect which we should do, which we should look into our research. And definitely, sir, I will look into the collaborative aspect. Yes, our university is interested in collaboration, and recently Balak have joined with you, and he also said to me that we can initiate Preeti, Balak both, and we can definitely work on this. And your previous faculty member Amit Goyal sir is the head now at this position, head of the department right now. So we can definitely work on this, sir, and it is very interesting because, as you said, there is a workshop to held tomorrow on zebrafish, and definitely some good aspect can be learned by the participants. So yes, sir, I'll definitely initiate and will connect to you in the future so that we can do the formalities and start doing something together. So thank you once again, Dr. Charan Singh, GD Gupta sir, Prabodh Sharma sir, for giving me this opportunity and sharing my research. And for me also, it was wonderful experience.
I congratulate you, sir, on getting CS number of donepezil conjugated.
Yeah.
Sir,
Thank you.
Use sir.
Thank you
And
so much.
you did the work in a smooth way and straightforward
Right, right.
quality presentation. So I thank you for sparing some time for the lecture and be part of the Ibro Spotted meeting and workshop. At the same time, I do thank Professor Prabodh Sharma to spare some time for the chairperson of the session.
Okay.
Thank you so much for both of you. And stay in touch, sir. We'll do something in the course of time. Thank you so much.
Thank you.
The last
Thank you. Thank you.
for the participants that they will have the workshop on day 3, that is tomorrow in Ibro Spotted meeting and workshop. So Dr. Atis will be taking care of the workshop along with her team. So stay in tune with the Ibro Spotted meeting and workshop. Thank you so much. See you soon.